Good morning, everyone. This is us again at Mamy, zielone, pojęcie podcast by Wrocław University of Environmental and Life Sciences. And the university is based in Wrocław, Poland. My name is Jowita Chojcan, and my guests today are researchers who both study fungi. And I don't know why, but my idea is that they're studying it because there are still plenty of new fungi species to be discovered. What's more, with every new fungi species, an amazing bioactive product, such as penicillin in the past, may be uncovered and then applied in the food industry, cosmetics, or medicine. So just to start with the two persons here, Dr. Ewa Szczepańska.
Good morning.
Hello, Ewa. She works as an assistant professor at Wrocław University of Environmental and Life Sciences. And she, in fact, graduated and was awarded her PhD also from this university, and that was in biotechnology. But she also completed studies in chemical technology at another university, at Wrocław University of Science and Technology. And also she studied production management at Wrocław University of Economics. Currently, she's pursuing another study. Wow, Eva, that's incredible. In the field of technology of cosmetic products. And her research focuses on producing bioactive natural products, especially aroma compounds, for potential applications in food and medicine industries. Since 2021, she has been the leader of the BioVan. Is that right?
Yes.
BioVan right. Project funded by the Leader 12 program by Poland's National Research and Development Center. And the aim of this project is to develop a biotechnological method of producing vanillin from food industry byproducts. So one more time, thank you for coming to the studio today. Dr. El-Sayed Ramadan Ali is a visiting professor at also Wrocław University of Environmental and Life Sciences. And his background is that he comes from Egypt. And he was awarded his PhD in applied microbiology at the Ain Shams University in Cairo. And before coming to Poland, he worked as an assistant professor of microbiology at the Nuclear Research Center at Egyptian Atomic Energy Authority in Cairo. His research also focuses on discovering and producing bioactive natural products for potential applications in various industries, including food and medicine. Over the last few years, his research interests extended to include endophytic fungi. Since October 2022, he's been carrying out the BioExplore project funded by the Polonez BIS program by National Science Center. And Dr. El-Sayed is a lead scientist behind the BioExplore project team. And together with Professor Filip Boratyński, who acts as the mentor in this project, they have created a larger group of researchers involved. And Dr. Szczepańska is one of the regular collaborators. So very good morning to you. And I'm really thrilled, actually, to have you in the studio today, because I really believe that fungi is an amazing area of science. So first things first, can one of you please explain the difference between fungi and mushrooms?
The kingdom of fungi is large, include mushrooms inside. And I have heard that in the Polish language, there is only one word describing all of them. But we need to differentiate. The fungal kingdom includes yeast. This is the tiniest organisms, could be unicellular, could be more, and mold. This could be filamentous, or more or less, and the mushrooms. Mushrooms are the visible thing that you can see and collect from the forest.
Which I'm sure a lot of people do.
Yeah.
Yes, especially in Poland, it's kind of fun activity.
Exactly. And I heard that this is getting more and more popular around the world. That, you know, maybe in the past it was mostly here in Eastern Europe, but now I think, Ola, you said that aside, I think, that, you know, scientists and ordinary people, everybody kind of looks into nature, into the wild to find, I don't know, good quality food. And I think mushrooms
it's very
would be a good
attractive
example.
and very popular food as a source of protein, and it is much cheaper than meat, for example, or other sources, and it's easy to grow it and maintain it. Actually, some people, I guess, have their own small farms and houses for growing mushrooms to eat.
And of course, there are also not only people, you know, ordinary people, individual people, but businesses. Yes, there are all these mushrooms that are farmed, which we can buy in the shops. But as you said, fungi or fungi, which should I say? I'm always really confused.
Both are correct.
Both are correct. Okay, great. So this is an amazing kingdom, and I love that. Even in science, we can say kingdom. That kind of, for me, makes this species very special. In fact, it's, I think, in general, it is agreed that it's between 2 to 5 million species, and yet only about 150,000 have so far been formally discovered and described. So how important is it to you that there is still so much more fungi to discover?
Fungi as organism, the whole fungi, you can say it has high diversity, and they can live everywhere. Actually, scientists have isolated fungi from the space.
Really?
Yes.
Oh my God.
And from the oceans, from the land, and my big interest from plants. So it has a very high diversity, and more than plants do.
Okay.
There is a lot of types of fungi. This gives it the importance as a source, as a living organism. A fungi could play a beneficial role in our life, and could be like a pathogen also. So we need to discover, we need to know about them as a source of bioactive materials, as a biotechnological player, like plant, and also as, I don't like the word enemy, but we need to.
But we have these pathogenic fungi, and that. But wouldn't that be correct if I said that some of these pathogenic fungi we can actually beat using something like penicillin, for example, which is again coming from fungi.
From fungi, yes.
So
yes, it's we can use fungicides, like compounds which kills fungi.
Fungicides, general term antifungal, and also it can come from fungi itself, because they are like any living organism. There is a competition.
Right.
Some groups can live in a mutual relationship. Some don't like. There is a term we call it antagonism. They don't like to be together, so they compete. Everybody produces its toxins to kill the other as a competition to survive and to have the land, the nutrient sources.
But why is it? Can you? I don't know. Maybe you can explain. Why is it that, as you said, the kingdom is so vast and diverse, and yet only so little has actually been formally analyzed and described? Is it because, I don't know, scientists did not treat fungi as an interesting area, or is it just because you can't, this vastness makes it almost impossible to know everything about it? Yeah,
this is actually the reason. Mycology, we call it. This is the science of studying fungi. It's started in the deep history, and scientists since then they have been working on it. But it is very vast, as you said. This is a huge diversity, and we can see it's uncountable. Not like plants. A visible plant, you can deal with it and can study. For fungi, it lives everywhere, so it is uncountable. We cannot say that over the planet there is a definite number, because we cannot cover the whole planet for research, for example. And coming back to the endophytic fungi,
which is the subject of
Yes.
the BioExplore project, which
Yes.
you are currently leading.
my main interest. This class of fungi live inside plant tissues, and they are totally different from pathogenic, that they infect the plant. And another group, we call it epiphytic. This live on the parts of the plant. Each plant, each tree, for example, if we have two trees standing together and from the same species,
Right.
each plant has its own fungi.
Wow.
So imagine how many plants we have over the planet.
How many? Yes, we definitely have a lot of tree
Yeah.
species. And so are you saying that they are all different because of the
Yes.
fungi that grow with them?
Yeah.
Right. Okay. Now, I'm really curious as to how did it happen, Syed, that, you as somebody coming from Egypt, that you have taken interest into Polish forests?
After my PhD, I had a project, a dream to pursue. I was looking for the right funding scheme, and for my good luck, I contacted Professor Filip Borotynski, and we tried with you, of course, submitting Marie Curie postdoctoral fellowship and other granting scheme until it was funded by NCN. Before coming here, and after the first attempt to submit to a Marie Curie grant, it was rejected. And so Professor Philippe told me could be more, it could be better to come to see the city, to see the country, to see the university, see the infrastructure. So you have a real picture about what will be. I came here to Wrocław in 2021. It was on November. And the first thing I have realized, this is the fascinating nature. A lot of green areas and trees, really. And coming back from Egypt, most of our landscape there is planted trees. We don't have this
naturally growing trees.
Wild nature, as I like to say it. So, taking my main interest about endophytic fungi.
Okay.
I have been isolating them from normal trees from Egypt, and looking to here a lot of wild, untouched, untaped, and searching the literature. Also, was no active research on targeting this specific class of fungi, endophytic fungi, living inside tissues of forest plants.
Right. So I see, yes. I see the attraction. And also we talked about it. I think you have like as a young, maybe first student and then young scientist, you have taken a particular interest in Marie Curie.
Yeah, I have been obsessed during my childhood by the person itself, the great scientist, Marie Curie, awarded 2 times Nobel Prize in 2 different disciplines, and discovered an element, polonium.
That's right.
Named it after Poland. I know a lot of details about not only her scientific career, but also about her private life. I see it as a legend, actually. And for the good coincidence, I came to Poland under Marie Curie action. Poland is biz.
That's true.
And also for the good coincidence, also, I live at a street named Mary Skłodowska-Curie. Under her name.
That's
also
incredible, isn't it? I mean, here is a person who has sort of been taking an interest into this wonderful scientist, and now, not only the program, but the street. Excellent. And Eva, I guess your interests are maybe slightly different, but did you go mushroom picking to the forest as a child. And
Yes.
has that kind of maybe, is that where your interest in natural things, naturally obtained substances such as flavorings come from?
As a child, I had such activity. I was going to forest picking mushrooms. It was like standard family Sunday.
Right. Except that you had to get up really
family.
early, so I hated that, I
And
have to say.
I especially like picking up, it's called in English saffron milk cup.
Okay.
In Polish, it will be rydz. It was my favorite mushroom. I knew where to find them, so I was always going to the special place where I could collect them. Interesting fact, the Latin name is Lactarius deliciousus. So it's so delicious.
So it's delicious. Yes. Wow, excellent.
But of course, as Said mentioned, fungi and mushrooms, it's the same kingdom, but they are completely different by appearance, especially by appearance, because mushroom, which we collect in forest, it's a different type. It's called Basidiomycota, to be exact.
Okay, in Latin, right.
Yeah, and the kind of fungi which I am studying, it's Ascomycota mainly, which is it's a different thing.
So there won't, let's say we've got your fungus delicious, if I may call it, and then so if you take that and analyze in the lab, that can be used for cooking, but for that kind of, for example, these flavorings that you are so particularly interested in, is that not a good candidate?
It can be, because you can grow it in laboratory, which I actually do that. You can do that, but it's harder. It takes more time than filamentous fungi. Filamentous fungi grows very fast. And Basidiomycota, mushrooms, they don't grow so fast. So they are a little tricky to work with.
Okay.
It's not simple structure like filamentous fungi. It requires more time, specific nutrients.
Yes,
you can say mushrooms is a bit near to the plants more than
it needs
the
cultivating
fungi.
proper,
Yes.
right.
And coming back to the term kingdom of fungi, to be 100% scientifically precise, there is two kingdoms, the animal kingdom and plant kingdom, and the microbes are under the plant kingdom. This is for the systematic point of view.
Right, okay. So the fact that people say that there is something like the fungi kingdom, they're not, well, they're kind of simplify
You can
that,
say
I guess.
as kind of simplification, but this is if you are talking about the systematic point of view.
Then there are only two kingdoms.
Yeah,
and
there
right.
is a sub-kingdom under the plant kingdom.
And unfortunately, with mushrooms, we experience edible ones.
Yes.
I love that fungus, delicious. And the toxic ones, yes, the poisonous ones. And I would say, would you agree, Eva, that let's say Polish people roughly know which one to pick? What's the awareness like?
Every year we hear about accidents, which unfortunately happens, that somebody cooked poisonous mushroom. And it needs experience, first of all, and a little awareness, right? To pay attention to the details, because some mushrooms are very similar, right? And only details deteriorates, determines
that one is edible,
exactly. These are just details,
Right. So, and what about our endophytic fungi that Syed specializes in? Can a normal person just go to the forest and collect them? Can they?
It's not that easy.
It's not easy, yes.
Because they live inside the plant tissues. First, you need to take them out. And as a plant part or a plant, I just said that there is a pathogenic, so you need to collect a healthy part. This is one thing. The second thing, you need to kill all the epiphytic, the microorganisms that live on the surface of the plant part itself. Because everywhere, in every surface, plant or a land or anything, there is microorganisms, bacteria, fungi, and so on. Right. So first, to grow them, as I mentioned before, that there is a competition between microorganisms. So you need to disinfect the surface and cut the plant part, so the fungus from inside start to protrude. And also the plant part, you need to put it on a sterile medium. We mean by medium, this is the food.
Okay.
Okay. Solid medium, this is like, let's say, a piece of cake for the fungus to
feed itself?
Yes,
to grow.
To grow. Yes. So it is not so easy for a normal person. It needs a lab and sterile conditions.
Right. And you said you came to Poland and you liked all this, green areas we've got.
Yes.
And so I guess I don't know, at some point you had a pretty good idea where to collect the fungi needed for your research. So where exactly did you go? And did you go somewhere here in the local area?
Yeah. Mostly I prefer the evergreen plants. I mean by evergreen, the higher plants, not shrubs, not grasses.
Right, so like trees.
Yes, like old trees.
Old trees.
Old trees have more interesting fungi because the age is so old. So it could have something rare, something that have never been isolated before. So I asked Professor Philippe about what kind of forest is here. I need the wildest one, the untouched one, and we selected a forest near Wrocław. Its name is Szczepańska.
Okay.
I guess it's 5 or 10 kilometers from Wrocław here. And we went there, and with the help of Professor Kasia Petejok from Department of Plant Protection at the university also, she helped us to identify the plant species.
Right.
So we collected samples from 12 plants.
Okay.
Most of them are leaves, if the plant has leaves, or needle.
Yes.
If the leaf have the needle shape, and bark, pieces of the bark, and twigs, these short branches. And we transported them to the laboratory at the same day, started working on the isolation.
So you went to Szczepański forest, and did you find all that diversity? So as you said, leaves, needles, that kind of bark that you were interested in?
The forest is almost untouched. This is like a community of grasses, higher plants. Also, you can see small animals. So it was very interesting.
Do you go back to that forest maybe just for fun, just to kind of
visit, of course.
To visit, to inhale fresh air and then right.
I in my personality, I have a connection with places. So if I visit a place, this kind of small relationship, I tend to visit one more time, spend some time there. Yes.
I don't know if Las Szczepański needs more advertising. But if it's not very popular, I guess everyone who's listening here today, maybe they should pay a visit. Do you recommend it as a nice kind of walk around and kind of a place where somebody who normally lives in the city can kind of have that closer contact with nature?
I guess if I can say this better than me, because I see all forests here are amazing.
Yeah, for Said, especially every such place.
All the landscape or the green view are amazing, actually.
Yeah, because for us, I guess it's like normal thing. Like every of us, I assume, went at least once in their life to the forest.
True.
And I guess for Said, it's something completely new. And
yes.
But I am happy for him. He found this so fascinating. It's actually very, very for me, it's also new that somebody can have such attitude towards forest.
Okay, I
because
see.
So
right.
for us, it's like normal thing.
Yeah, that's true.
Yeah, so I find this quite interesting that somebody can have approach.
I'm looking to the forest from the point of view of the vast diversity. There is a lot of everything you can see, and also coming back to the term discovery, if you want to discover something, you have to go to something and touch it. You would be like the first person, perhaps the second person, to start discovering what's there. So there is a high possibility to find something new.
Okay, interesting. Have you ever thought about that, Eva? I guess most of us, I suppose you were these... you were looking, you had a purpose to go to the forest. You wanted to find your favorite mushroom. But some people, I guess, they just go, to get some fresh air. And yes, your approach is kind of interesting and very different. So actually, did you discover any new fungus in that Mokrzański forest? And if so, what kind of magic powers does it have?
On the BioExplore project, we isolated good number of fungi, 46 fungi, and
46?
Yes.
Wow.
And from 12 plant species, different plant species. Some of them we started to identify genetically and morphologically. This is from the scientific point of view. To name something, you have to identify it first, basing on their importance. Of course, I will not identify all the isolated fungi because this is a lot of work, so we focus only on the most interesting ones.
And how do you make this decision? This is interesting. This isn't interesting.
Is not. Okay. First, the main goal of the BioExplore is searching for new carotenoids and other pigments. Carotenoids, this is a class of compounds, is very important to us. And the industry for production of carotenoids is very prosperous because it is sensitive compounds. And I can give you a simple example like beta-carotene. This is a very important compound. So we made a screening process to see if there is carotenoids inside this. I will try to be very simple. Okay, to see if they contain carotenoids and pigments or not. Of course, for pigment, you can see it is a colored fungus or colorless or
under the
white one.
microscope, it has...
It's not even, you don't need even microscope, you just look at the petri dish.
We grow it on normal petri dish, so you can see it is colored, has a red color, black color, yellow, and so on. And this is for the pigment itself. For carotenoids, it could be there is carotenoids and a color that is not visible to the eye, and after extraction and detection was some techniques like liquid chromatography, mass. You can see what is there by comparison with known standard of these carotenoids.
Okay.
And I can say right now we have a very interesting fungi. One of them produces beta-carotene, and this is from the biotechnological point of view is very important.
Tell me about this beta-carotene a little bit more. Where is it? I mean, which industry uses it?
Beta-carotene is... there is no industry use it. They produce it as a food supplement.
Okay.
It has a lot of medical applications, medical uses as a food supplement for the immunity, for the health.
It has antioxidant activity.
Do they actually do they prepare like supplements that people take?
Yes.
In case there is a
from
problem.
Beta-carotene and extraction beta-carotene, or let's say that's the natural one, because there is a global trend to move to natural sources instead of synthetic ones. Because synthetic ones, from industry point of view, it could be more costly. And its production process requires certain conditions like temperature or toxic solvents, so it has an impact on the environment. So having natural products like beta-carotene from natural sources has a good impact in terms of sustainability and the production cost itself. Beta-carotene is so important compound, and It's production like most of natural products from other sources, but finding a fungus, and if you just search the literature, and I mean by literature the published research in this area, you will find this is a rare research papers on using fungi to produce beta-carotene. And the fungus is quite interesting, and I will not say the name right now,
Okay.
Because we're still working on it, and it could be like patented. Another important and interesting, two fungi, not only one, they produce melanin. Melanin is a quite important compound because it's a pigment you can find in our hair, skin, and right now it has a lot of biotechnological applications and industrial also.
Okay.
It's as a black pigment, with this importance, I call it like a black gold.
Because what? Because it
Because
could
it's-
bring you
yeah.
so much money at the end.
Yeah, because of
well, I don't know about you, but people who
because
will
of
use it.
the industrial high significance as for this compound as a pigment, it has a lot of interesting properties about electrical properties, magnetic properties, and as a color also a black color could be used as a dye.
As a dye,
Yeah.
yes.
And some research is starting to work on the biological activities of melanin itself. So it is a pigment and at the same time has biological activities like anti-cancer effects.
Right. Okay. So it goes into it sort of when it interacts with another, let's say, substance, then it can, like you said, it can help people with cancer
if
treatments.
I can add, actually, most of people think like melanin. It's as you mentioned, it's
The black one.
But we can actually divide melanin into two kinds. One is pheomelanin, which is reddish, yellowish. And the second one is eumelanin, which is brownish, blackish, I would say. That's why people have blonde hair, right? They have brown hair, red hair also.
Because melanin is... this is due to melanins, let's say.
The kind of melanin
pigment.
which we have in our hair determines what kind of color we have. And also, as Said mentioned, it has some biological activities.
Yes. And one of the most interesting activities also its ability to be a radioprotectant.
Yes.
Against radiation, against UV.
That's why we get
radiation.
tanned, for example.
So a lot of cosmetics rely on it as a sunscreen for protection from...
It protects our skin from
your blood, from the sun.
That's right.
On top of that, and this is very significant thing, it has a radiation protection against ionizing radiation. I mean by ionizing radiation, the most energetic one that could come from a nuclear pump, for example.
Oh, okay.
Gamma radiation, for example.
Right.
At specific doses, not the highest one, to be scientifically correct.
Sure. So it seems very versatile.
Yes.
Going from
medicine to food to cosmetics to industry in general. Also, melanin could be used for solid-state batteries. It has...
Say it again, for...
batteries.
Batteries, okay, right.
Generally.
So, hold on. It has all these wonderful properties, but I guess, like with all biotechnology, it needs a specific process. So what's happening? There's your research that says, I have a fungus here.
Produces melanin.
Which produces melanin.
Yeah.
And is it possible to kind of take it to some kind of next level and develop, a process that can be used by industry?
Let me talk first about the problem of natural melanin. Production natural melanin. Here we are talking about natural because there is a lot of patents and research paper about synthetic one. Here we need to replace the synthetic by natural. This is the global trend to go to natural products because this is safer for the human being and the environment as well. The problem of producing natural melanin from its sources, because its extraction of melanin, the process itself is costly.
Right.
So you need to find a simple source. If we can present to the scientific community, to introduce to the scientific community something like a fungus or bacterium or yeast, normally replacing plants or animals as a sources with microbes is in terms of sustainability, it's okay, a very good thing. And in terms of cost also, imagine, for example, growing a plant to produce a specific chemical. And here I'm not talking only about melanin, I'm talking generally about planting 1 hectare of land and waiting for 3 or 4 months as the whole season for the plant to grow and harvest and extract and take your active material. Compare this process with producing it in a lab, in a scale fermenter, for example. And you can have from the microbe, whatever it be, the same amount.
And quicker, I imagine.
Quicker, simpler, safer. In terms of environmental protection, it's okay, because for plants you will have agricultural residues after extraction. Extraction process is costly and uses toxic chemicals, so it has a negative effect on the environment. Okay. So using biotechnological routes is more favorable right now.
And also space-wise, right? Because you have to take hectares of the field to obtain, for example, 1 kilogram of some compound.
And we can find.
Here we have just 1 container, 1 bioreactor. We can do the same amount.
If we can find a fungus, for example, endophytic fungus, that produces melanin in a significant amount, it will be of high interest to industry.
Right. Okay, great.
And this is, we are presenting the starting organism. After that, there is a lot of processes. The organism, the machinery for producing could be modified by genetic engineering and other tools to make it hyper producer. For example, if we start with like 1 milligram per liter melanin at the yield, after the genetic determination and then optimization of the whole process, it could be intensified and it could be satisfied for the industry that they use the fungus itself as a source for melanin.
And is that going to take a long time to actually get to that level?
Research, of course. Research is time and effort.
Well, I realize that, but I'm just thinking, you know, if you have a stable process on the lab scale, then going, Eva, I guess you maybe have a little bit more experience with that.
Yeah, because as you mentioned in the beginning, my project's goal is to obtain vanillin. And I started also from lab scale, of course, to do screening, to find fungi which will produce vanillin. And now I am at the last stage of my project, which aim is to construct bioreactor in a bigger scale, let's say, not industrial scale, of course, but much bigger than in Erlenmeyer flask, small ones. And it's very challenging, I have to admit. It's very challenging because sometimes, you know, you discover some process, some biotransformation, which works very well in laboratory scale.
Small scale.
And you want, of course, do more research on this compound. You want to do bigger scale, to obtain more of this compound, to do some purification, to do more analysis. And it doesn't work like that. Just one
upscaling process.
Upscaling is very challenging.
The process itself is challenging.
Yes.
Upscaling, moving from the lab scale to industrial scale, or let's say 5 liters. It's not industrial scale, it's bigger scale, challenging. And imagine moving to the big scale industrial fermenter.
Of course, it's possible because it's already happening. There are companies producing, for example, citric acid.
It's
right?
challenging, but it's not impossible. It needs joint efforts between the researcher and the industry.
Well, that kind of, all what you've just said brings me to the next question. I wanted to ask about how you see this joint effort going forward. Do you, for example, Syed is well aware, and I'm sure maybe you have the same requirements, by the LEADER program. But for example, if your project is funded by any kind of EU funding program, then you've got to set yourself really ambitious goals in terms of economic impacts of your project and societal impacts possibly
and I guess
as well.
In our case, it was like very one of the most important factors, this environmental issues, let's say.
Because we are working on the area of natural products. This is quite interesting, and it's on the top of European Union goals. Not only to move from the synthetic ones to natural ones, but also to sustain the whole process of circular economy, because fungi as living organisms has the ability to grow over the wastes, like wastes from agro-industrial wastes, food residues, for food wastes also. So we can close the loop.
Right. Oh, that's very interesting. So it makes sense. And I think, as you said, this is the priority now to try and close that loop.
And, one more important thing, I guess, there is also customer demand on natural products.
Absolutely.
That's why industry is interested in that, because
the customer preferences moving from a synthetic one to natural. And of course, natural ones are expensive. Putting scientific efforts in this area may contribute to lowering the price of this natural product. So there will be a huge competition. So the consumer will take directly the natural one. It will be the same price as
the synthetic one.
If I can give you an example, vanillin, of course, like in my case, natural vanillin, like actually extracted from plant, cost $4,000 per kilogram. $4,000.
How much of it?
1 kilogram.
1 kilogram. Okay,
yes.
Sorry. Yes,
no problem.
1 kilogram.
So,
$4,000. Wow.
And it's, of course, it's from plants, so there are environmental factors impacting on the process, right? There can be some environmental disaster, for example, right? There are a lot of factors which can make this process less economical.
Seasonal change also, geographical changes.
Some disease, plant disease. Also.
A plant could be planted in a specific area of the world and not other area. It requires some climate conditions.
Vanilla planifolia, the plant.
And that doesn't grow in Europe, does it?
No, it's Madagascar. It's like very territory-specific plant. That's why it's so expensive. And now we have synthetic one, which we, on daily basis, we eat, to be honest, because it's in ice creams, in creams, this vanillin sugar, right?
It's always added to chocolate.
Yeah, to chocolate.
It's always vanilla
cake.
chocolate. When you read the list of ingredients, vanillin is really, you saying
it's very popular to kids, I guess, and
so we consume quite a lot of that synthetic version, I would say.
But the thing is, synthetic version is obtained from petroleum.
Really? Oh my God. Should I stop eating all available chocolate?
No, it's
right
like
now?
I know it like sounds scary. Of course, it goes through some special chemical synthesis. So the last product, it's vanillin, which is, I hope, purified properly. But the fact it's obtained from like kind of compounds, it's a little like not. For customer point of view, it's not pleasant to know, right? That we eat something which is actually from petroleum. And that's why in my project, I am trying to do similar thing as Syed, but different approach. I want to produce vanillin, which will be natural by European law, because it will be obtained biotechnologically.
Yeah. Using a fungus?
Yes, using fungi, which has grass status. Grass status, it's also very essential in biotechnological process. Grass, it's the abbreviation from generally recognized as safe, which means
so
like we can eat it. We can eat it. It doesn't produce any toxins, right? It's recognized as safe. So there is no harm for our body if we will eat. So fungi are included in this process. We are sure generally this whole process is safe. So the product, it's also safe.
And have you had interest from industry?
Yes.
Are you working with maybe an industrial partner?
I have collaboration with 2 companies right now. And especially one now, it's actually the end of the project. So we are preparing for the last stages. And as we mentioned, of course, the laboratory scale and industrial scale, it will be for sure challenging.
Challenging, but it's not impossible. That's joining the effort.
Yeah, it's not like impossible. It's possible, but with proper resources. Yes, why not?
Here in the lab, we are working on a term, we call it upstream processing. There in industry, they are working on downstream processing, taking the product, purify and make it as a final product and sell it to the customer. And coming back to the cooperation between industry and academia generally, in most of industries tend to have their own R&D.
Yes.
Unless your work is really significant, scientifically important, have a great discovery, it will attract the industry. They will come to you.
And Eva, I guess that was your case, wasn't it?
Yes, in both
they
cases.
Came to you.
Actually, company reaches to me and they ask, they were interested in my area of my field of research, and they ask if I can propose for them some solution for their problems. So I agreed to help them, and now we are collaborating. So it came. So,
did it come as a little bit of a surprise, or did you?
Yeah,
were you doing it with this objective? Come on, I want to
actually,
make them
it was
interested
actually,
in my work.
Coincidence, because I just defended my PhD thesis in this matter which they were interested in. So yes. It was
good timing as well.
Yes, very good timing.
I wouldn't call it coincidence. I'm sorry, it was just timing. You timed it just right, Eva. Now, we talked about these impacts that, your research might have, and hopefully taking some of what you're discovering into the market. So let's stay on this subject a little bit. I think there was also another fungus, Syed, something that a fungus that produces a strongly red pigment.
Yes.
And I guess you've sort of shared with me before an idea that there might be a new product on the horizon. Will you kindly tell your story on that subject, please?
It's a well-known fungus called monascus. It's actually used from the deep history in food. And China is growing the monascus over rice to have red rice. And it's already a product in the market. You can buy red rice. This is normal rice and monascus growing on it, producing the pigment. A second direction of the BioExplore project about exploring. We already started working on that topic for production of monascus pigments on 2021. And we continued after the project start. Currently, we are evaluating different food wastes. So we will grow the fungus on food waste. Let's say bread residues.
Okay. So basically what's left?
Leftovers.
What's leftovers from baking?
Yes. So we will grow the fungus over it, and the fungus will produce monascus pigments. There are 3 types of pigments: red, orange, yellow, and the most important one from industrial point of view is the red, the most attractive one. So we are focusing on the red pigment, and the fungus is safe. And the pigment, after extraction and purification, we will try to use it as a color for production of red bread. Hopefully it will be attractive.
Well, exactly. Because I was thinking about that. In fact, I asked my parents. I said, Mom, would you be interested in trying red bread? you know what? I think she was a little bit like, ooh, I don't know.
Maybe for adults it's not attractive, but for children, for example,
be, yes.
like you can prepare an interesting meal, like from the like
visual point of view. Yes.
From the psychological point of view, it is negotiable. You can say if you present an idea and you hear no, so it's absolutely no. But if someone will think, let me try. I can see. So there could be.
You guys are tough negotiators in your part of the world. It's that's just speaking from a tourist experience. If you just, it's not a definite no. They'll try.
This is what I learned also. Like, it's
this is
very good approach.
Yeah, from our research.
But
we are dealing with facts.
Right. I think kids, they might. I mean, there are these crazy.
There is
because
also
red color is very attractive to kids.
Yes, that's true.
Unusual. And what I want to add also, because this red pigment can be also kind of preservative, like antioxidant, something which will prevent bread from
going bad or going stale.
The pigment itself has bioactivity. As antimicrobial, it kills bacteria and microorganisms.
Actually, that's creates good question why actually plants produce like pigments. It's actually to protect them from radiation,
it's not only
from
coloring
pathogens. It's not
coloring
just
compound. It also protects. And also from our research, we evaluated the pigment extracted from this monascus. It has anti-cancer potential, and it has antioxidant and antimicrobial as well. And coming back to the monascus work, we have developed a mutant. I mean here mutant. Let's clarify the term.
Scary, but,
mutation. You can modify the fungus by changing its genetic material, DNA, for example, and direct it to producing more or less something you want. If it produces something good, desirable, so you will intensify. And
so maybe those antioxidant properties?
Yeah.
Is that what you're saying? That you
the production itself, the titer, the yield. Also, you can direct the mutation to get rid of mycotoxin or something.
Okay.
So the mutant or a daughter from the parent. Here we have the organism, the wild one. This is the parent. And the daughter, this is after mutation. This is the mutant. We developed this mutant using gamma radiation and collaboration with my home institution in Egypt, because the facility is available there to use gamma radiation. It's a powerful radiation and it targets the DNA. And the mutant has improved productivity of red pigment. And we used it to produce pigments, and we identified, we used it to develop a process. It could be attractive for industry on a scale of, I don't want to say the term, immobilization technique. This is
okay. Sounds like very confusing.
This is a complicated term, but this is kind of biotechnological methods preferable for industry. It consists of, you have the fermenter and your fungus. This is the biomass. You are growing it on a support. So you can easily separate the end product. You have the biomass here and the end product here. This is preferable for industry because whole fermenter have like inside the rotor and the stirrer inside. The biomass stick on the walls, on these things, block the bumps. So it is preferable for industry to have this kind of support. You grow your microorganism on a support. So the whole biomass could be collected easily.
Okay.
Okay.
All right. So I'm just thinking, one thing, because you said this, that it's very important that whatever you do has this, not certification, but the GRAS thing, it's considered safe.
Genetically recognized as
safe. So is this, kind of creating that daughter with this gamma radiation, is that considered safe?
Yes, because the radiation effect ends after the irradiation process ends. So there is no any harmful effect from the radiation. The radiation targets the organism itself and modify the organism. After that, the effect of gamma radiation ends. So you have a modified microorganism.
Yeah,
it's kind of mutagenesis.
Yeah, exactly what I wanted to say. It sounds bad when you say mutant, but mutants are naturally, like they naturally occurred in nature, like bacteria, fungi, yeast.
It's like,
they are
you can say generally
mutating all the time.
The term breeders. We are breeding the microorganisms, like having good one, making it better.
Kind of improving, updating.
Yes.
Okay. Well, now I feel a bit safe, more safe about this mutation.
And this is
stuff.
the point. We have a publication right now. It's under consideration. I would like to say about this red pigment.
Okay.
This red pigment and the mutant thing for production of high levels of red pigment. This is another thing from using the monoscous pigment for food wastes and the red bread.
Okay.
And coming back to melanin, I would like to say the two producers of melanin, till now, they are new fungi. They could be novel species. We are working on it. We started with identifying fungi. We started with using the recommended things and the easiest because everything costs. We started with the morphology and we can't identify it under a microscope or as a colony. It's something new. We moved to the genetic level. It is kind of deterministic. We use, I will try to simplify something like targeting specific gene sequence in the DNA of a fungus and compare it with the database
of existing microorganisms.
Yeah, the whole organisms, animals, human and plants and microorganisms. All this is a worldwide database in CBI. And after making these comparisons, we can't identify it. And actually, we are working with Professor Tomek Shawa from Department of Genetic Engineering. He is a professor. So we decided to move to the top level. We will make a whole genome sequence. It is costly, but it is like a deterministic thing to say this is something new or not.
I see. So you will follow, like your hero, Maria Curie, possibly, which I wish you all the best of luck with that.
If my project will contribute to describing a novel species, I will be very satisfied.
I can imagine. Wow. Okay. And BioExplore project, I remember I read a lot about it. You have a large group of collaborators there. Some of them are internally, like you mentioned, people within the university who have all contributed, but also there are some external guys. Can you just, briefly tell us what was the role of this larger group? How did it all come together, for the benefit of the project?
Together with Professor Philippe and taking his advantages of his wide network, not only on the national level and also international level. And also my home institution, I would like to connect it to benefit from the work here. We built a large team, or a big team. From here, from the mentor, and we have a co-PI and 2 PhD students. They are mainly employed under the project. The others are collaborators. We are collaborating with the medical university, Wrocław Medical University.
Okay, so another university.
Yeah, Professor Jowita. Because we have group of fungi, the whole group of fungi, endophytic fungi, we isolated. We are trying to study its anti-cancer potential.
And for this, you need the expertise of the people from the medical university.
They have the required infrastructure and material and experience also.
Okay.
So we made extracts from these and we collaborate with them to test against different kinds of cancers.
Right.
Hopefully to identify within the extract. I mean by extract, could be a huge number of compounds, not only one compound.
Right.
So it requires next steps to separate individual compounds and know which one is responsible, or which two or which group is responsible for this anti-cancer activity. And another interesting thing, we are collaborating with University of Łódź. Professor Joanna. Professor Joanna is specialized in vitro assays. In vitro assays, we mean by in vitro assays that we are testing something from a living source on bench top, not inside the body.
Because there are like two kinds
of the body.
Because if we say there is an enzyme in the human body that is responsible for a disease, and we test it in vitro, you need to differentiate, because it sounds like fungi can heal people with diseases. No, we are in the earliest stage in vitro assays. This is testing these extracts against different kinds of enzymes. And with Professor Joanna, we tested all the extracts against something like monoamine oxidase inhibitor.
Okay, that's very technical, I think.
Monoamine oxidase A inhibitor. This enzyme is responsible for a lot kind of neurodegenerative diseases like Alzheimer. We tested also against acetylcholinesterase. Its uncontrolled levels in the human body and the brain specifically cause Alzheimer, because cholinesterases, acetyl and butyryl, these are responsible for maintaining the neurotransmitter acetylcholine.
If it's out of balance, then a person gets ill, and if it's working well, then everything is fine.
So, in human, people with Alzheimer, it's uncontrolled level of these enzymes need to be adjusted. So they need to take external thing to adjust the levels of these enzymes.
Right. So hence the big group, because you're looking at various aspects of how fungi can
taking the advantage of this group of fungi. It could be new. The possibility of finding something new is high. It's not simple work, but I usually plan work for years. We can start now, and the work can take along. It opens a lot of opportunities for collaboration with our people.
And also, if I remember correctly, the whole idea of Polonez BIS was to kind of give you, as a lead scientist, some new skills, and not only research skills, but also skills in management, in, I suppose, research design. Is it the same for a leader?
Yes.
So
quite
now,
similar.
Now both of you are, let's say, you coming towards the final stages of both of these projects. Do you feel like a lot stronger and a lot more experienced having done the work that's already been done? Eva,
especially I would say in the case of experience, right? Because it is my first so big project. I am managing now like 5 people team, so it's quite challenging also. I am trying to learn also, because as I mentioned, it's something new for me. So a lot of experience I have gained, I have to admit, not only in the case of managing people, but also like managing studies, also like to plan things. And on mostly every step there were some problems. So also like kind of like risk management was involved.
They always ask you, don't they, in every application?
So I was
think of risks.
What is your risk plan?
Like financial things also. So my study at University of Economics kind of helped me. It was helpful actually, because I could use some knowledge from this study. But like it's something completely new. I have to admit. But I am grateful. I have this opportunity because not everybody can have it, to be honest.
Do you feel that knowing what can go wrong in the project, you can then write better applications for future projects?
Yes, in the future I will know what to pay attention, especially like what kind of aspects of project should I pay more attention. Because like, we learn on mistakes also. It's not like everything goes perfectly. But this learning on mistakes, on problems, gives actually the most here.
And for you, Syed, do you feel that the future applications will be easier to write?
All the time it's challenging.
Question,
yes.
I know, sorry.
If you have an idea, you need to sell it good.
Sure.
Because it's from your point of view, it's a great idea, and I can pursue. But other people, reviewers and experts, have different views to your work, how you will do it. And coming back to the Polonaise piece, the program for management skills, yes, this is the first project. I'm the PI. And it was kind of challenging in the first months. Managing a huge group, because it's not only those people I have described, they're also a group of students are working.
Okay.
Some of them are undergraduate students, and usually I pay attention to students. This is the coming generation. They need to learn, especially if they are interested. They want to learn. They want to have something new. So being with them in the lab, teaching, showing, and giving them tasks to perform. All that is like challenging, especially coming from different culture, different language, different weather.
Even weather is involved.
Was it hard for you to like adjust to our, I don't know, gray November and December?
Now I am adapted.
Right.
So I can feel like a Polish person.
How long did that take?
Not so much. 1.5 years.
That's pretty good, actually, don't you think, Emma?
Yes.
And right now, also management skills. This is something new for me. To have a management skills, financial and managing tasks and work packages. Because the BioExplore project is kind of broad, have different directions.
We talked about all of these things that you aim, you wanted to...
Different directions.
These pigments, biocarotenes, some other things. And you're right. So this was probably hard to coordinate.
It's not so hard because I love my job, basically. So I I enjoy my work.
Sure.
Actually. So moving between these directions, it needs to be organized, giving specific tasks to the people. Everyone knows what he will do, and you can follow up with him besides your actual work in the lab.
Yes, I guess these soft skills are also crucial. Like not only knowledge, experience, but also your character, also your temperament. Because not everybody can be leaders, for example, right? It needs some... True.
And also Polonaise base gave me the opportunity for internationalization. Being here in Europe for the first time, going to conferences, international conferences, dealing with and having a network with European researchers, recognized researchers. And within the activities of the bioactive group led by Professor Filip Brotynski, he is doing great work about inviting a lead scientist every 3 months or something like that to give a lecture here at the university.
Right.
And actually, the last one was very interesting because he invited a professor from University of Vienna. She is a well-recognized scientist in the field of natural products. And when I attended the lecture, I thanked her so much and him for sure. And professor, I want to talk with you. I I want to collaborate with you. I have a lot of things to do with you. And she's okay. I'm very open. And literally, she visited the lab and we talked. And right now we have a collaboration.
Excellent.
Taking the advantage of her infrastructure there, equipment, and then when we send samples, we meet online, we talk to discuss the results and so on.
Great. So that is, I guess, something that Polonus-based people should be happy about, because that was the whole idea, to kind of bring international scientists into Poland and then letting them grow and also, kind of work with different scientific groups.
The Polonus Peace program right now is you can say it's modified than the previous ones, and right now it offers a lot of opportunities. There is a secondment also in industry for at least 2 months.
And hold on a second, a secondment is a term maybe that not everyone's familiar with. You basically spend time outside of the university.
Yes. In industry.
Yes.
And they put a rule about that. You cannot make a secondment in a research institution. It must be industry, a company. You need to go there to see how's it going in industry,
right?
To have an idea. I see this is a very interesting opportunity for me, for example, to go to an industry, whatever it is, big company or a startup, to have an idea how to move the ideas from lab and put it in a startup or an industry, how to sell it.
Yes, I guess that is you spoke about those challenges, so you could see from their perspective once you're there.
Exactly.
As long as you stay in the lab, you might not really...
Exactly. As a researchers, we are just not aware. We don't have contact. We don't have experience in the field of industry, which has a little different rules. Would say I guess this exchanging we give some kind of knowledge, they give us their kind of knowledge, completely different. We can together create something good.
Excellent. And if we're talking about the good points of the Polonus Peace program, if you remember, we had to have a communication strategy as well as part of the proposal. And this is something I always find researchers struggle with a little bit. So how did this turn out for you? Of course, we're sitting here. So, clearly something is working correctly. But would you have some advice maybe now that you are at the end, you're almost at the end of your project, how to approach this side of, when you think of a new project? Do you feel now a little bit more competent in terms of the, how to plan the communication strategy for it?
Yeah, of course. It was for me totally new, having a communication plan to, let's say, to the general public. Because communication to other scientists is so easy. I can give a lecture, but for the public, it's challenging. You need to simplify as much as you can. So young people, kids, children, and adults also can understand what are you saying. We went through that with the help of Department of Promotion of the university. Really, I appreciate their help. At the start of the project, I had an interview, and it was shared with many websites in Poland here. And few months ago, we had another interview about the dissemination.
The progress.
Yes, the progress, what happened, and which results interesting. And it attracted an international magazine called The Microbiologist. This is a well-recognized one in the field, supported by Applied Microbiology International. This is international organization for microbiologists. Besides a lot of activities, really, I am happy with.
This Naturalist Day. Yes,
the Naturalist Day.
We had that in last week, was it, or 2 weeks ago?
2 weeks.
2 weeks ago.
It was an open day for people here in Wrocław to come and see, and the main topic was fungi. It was very interesting, different topic. About endophytic, we have a tent, and we showed people the colors, melanin, and this is the fungus produced, and this is monascus, and this is the color, and here's under microscope, it looked like that, and this is petri dish. And There was other tents. I have been noticing also some people from a faculty of animal science, as I remember, presenting pathogenic microbes, pathogenic fungi, in particular to animals, and another tent was adjacent to us, presenting edible mushrooms. It was a great day with a lot of activities. Really, it was very interesting.
And did you enjoy, actually doing it, being involved in maybe preparing something, I don't know what kind of, whether speech or a presentation or something in a language that maybe is not like so, but
for me,
very scientific.
approaching general public, this is something new. I have been only the general public in my previous life. Previous life. That's like,
but we know what you mean. But when you worked in Egypt, I suppose
it was
the
like
previous stages of your career.
All the general people are like my family and their neighbors, those who are interested. What are you doing? This stuff. But for a large audience, like the population of Wrocław, for example, this is something new and very interesting.
Okay, great.
Yeah.
So let's hope that now people have a lot more knowledge about endophytic fungi and also about the edible types and the toxic ones. So do you agree with maybe something that I feel is happening right now, that there is some kind of growing fascination with all things fungi? What would you say about that? Do you agree that we can say that something like this exists in the world?
Fungi is a significant player on the scene right now, I guess. Because from the deep history, people used to use fungi. I will give you a very simple example using the bakery yeast. This is a fungus, a yeast class of fungi.
True.
So fungi are very near to us.
And also cheese, production of cheese.
Yes, production, Roquefort cheese. This is very famous.
Okay,
it uses Penicillium roqueforti.
Yeah.
Actually, for making Roquefort cheese, it's named after the fungus, or the fungus named after the cheese. Penicillium roqueforti, and also another type of cheese uses also penicillin. Penicillin was the first producer of antibiotic.
Yes, exactly. And that was like a mold, I guess, that was discovered sort of by accident?
Yes. The scientist Alexander Fleming was working on a bacterium, and he put the petri dish with the bacterium. He was trying to find compound or substance to kill that bacteria, and he lifted it. On the next day, I guess he didn't want to wash the plates, and he left it like that.
It was, I guess, kind of messy.
For a few days. He saw the mold, like a small ring, and around it, the bacterium was like a clear zone. The bacteria was killed.
By that mold?
Yeah, by that mold. So the bacterium was like growing on the edges, but near the fungus, there was no bacteria. So he guessed, what happened? Why there is no bacteria here? And from that, it gave him an insight to go what that fungus inside or produce. And after that, he isolated this compound and named it after the fungus Penicillium.
Right. So these are, a great example of things that happened in the past. But I think you both told me about some programs being available, I don't know, on streaming platforms such as Netflix. And that is also about fungi?
It was a serial about a fungus infected an ant.
Yeah, cordyceps, it's called. It's kind of fungi which uses insects to spread, like in the environment.
The term we call it entomopathogenic. This is insect pathogenic, the fungi that infect insects. And that fungus was kind of advanced. It's not only infected to eat the body or as a food. No, it controlled the brain of the ant.
Right.
It's incredible.
It is.
It's kind of science fiction because right now there is no proof that a fungus can infect and control the brain, or they are like all the creatures.
That's
but
true.
If they have a mind or they can think or not, no one knows.
Wow. I think I read something funny that they're quite particular. Some of the, fungus species, they, like you said, they need that particular brain to survive and multiply possibly. But also, I think some of them grow on the hair of I think it's cockroaches or something horrible like that, and this is all they need. They that particular fungus seems to rely on something that I find disgusting.
There is a lot of things. There are parasites. We call it obligate parasites. They grow only on a specific host. And others are like saprophytic. They can grow on dead materials like woods or something else. And other are like they can live in the soil, plants like normal growing. They don't need a specific host.
And I guess for those people who may be interested in exploring this a little bit further, I've come across kombucha kits. So you actually get definitely get the fungus and possibly like a starting liquid or something, and then you can see what it does to the tea and what kind of what kind of drink it will produce. Have you maybe had any experience in making it?
I tried. My friend made this kind of tea, and it has interesting flavor. You can feel actually it's fermented but
food.
In the good way. But for example, beer, it's also a fermented product. We drink that, so for sure it has some like probiotic properties.
Yes.
Right, improves
as a source of protein also.
Protein also, yes. Some like antioxidants also.
Yeah, it has a good nutritional values as replacing it with the not or the bad like drinks, for example. Yeah, this is something natural.
That's right.
When it comes to natural product, you will have a lot of benefits.
So it's a good alternative, and I guess if you
at least you will get rid of the harmful effect of other
things, and plus you can actually see for yourself. Yes,
you can kind of feel like scientists, growing.
Exactly. So finally, guys, because I think it's about time, we've got to wrap this up in a minute. Can you tell me, looking forward for the both of you, are there any unexplored areas of microbial biotechnology that you are, both interested in and would like to get into and explore?
Yeah, for us, I guess we will stay in this fungi area because it's a lot to explore. A lot.
It's still unexplored. There is a lot to do right now. You are searching for like a treasure hunter. Once you found one, of course, you will not be stopped. You will look for the second one, the third one, that, and so on. And my recent work is using this class of microorganisms, endophytic fungi, as eco-friendly platforms for the synthesis of nanomaterials. This is something trending, new, to use microbes to synthesize nanomaterials instead of chemical synthesis or physical synthesis. The science itself is still in its infancy because we cannot guarantee the successfulness of the process fusing microbes for nanomaterials, especially at high scale. You can do it using physical or other or chemical conditions. On big scale, you can synthesize, but using microbes, the science is still in its beginning.
Okay, sounds interesting. And you love your fungi?
Yes, of course. I work with fungi for I would say 12 years already. So it's I know them very well.
Yeah.
But there is a lot to discover. And one more thing, also this, what I mentioned before, this customer demand for natural products. It's still going. We have a lot of job to do in this field because the flavorings, the aroma agents, mostly they are synthetic, right? And it's good to find this natural alternatives for them.
Yeah, for example, those pigments, we are working on their production from Bioexplore and using new direction like endophytic fungi as a new source This is the point of novelty of the work, tapping the potential of endophytic fungi from forest plants. This could be exploited in many directions. You can target those class of microorganisms, endophytic fungi from forest plants, for production of aroma compounds, for example. This is a point of
also
novelty.
In natural medicine, we use plants, and maybe fungi can be kind of alternative for natural.
I don't want to say argument, but there is two theories about some research reported that endophytic fungi gain the ability of production of phytochemicals of their host. This tree produce X compound, and this X compound is a phytochemical, and it's important. Some reports reported that the fungus isolated from inside this plant produce the same phytochemicals.
Okay.
So it could be an alternative, or it could be replaced by the plant. Growing a plant is not easy, like growing a fungus. Fungus inside the lab or a fermenter, it require nutritional conditions is very cheap. A medium, the food for fungi, is so cheap. Not like growing a plant, for example.
And you said that medium can be helping with this closing the loop.
Yeah.
Yes.
So any kind of food production waste. Or
I also wanted to
from
mention.
Agriculture, from industry, also certain types of these residues could be used as a food for fungi.
Yeah, this I also wanted to use as an argument, because fungi are like the best tolerant for such.
In comparison with bacteria, for example, we can grow fungi under technology like the term is complicated. It's solid state fermentation. It has this fascinating ability more than bacteria. Bacteria need specific conditions for growing.
And fungi, like in comparison to yeast and bacteria, they have this ability to produce filamentous, which goes deep in the, like they spread very easily, very fast.
Typically, if you lift a piece of bread for a few days, you will see the mold.
Yes,
and
without anything, just a humid atmosphere, and the bread itself contains the nutrient. So they're growing.
We can even see on the walls they're growing. Like they produce vast amount of enzymes also, which help them to.
They considered like the best biotechnological agent in comparison with other microorganisms in terms of growing, scaling up, tolerance.
Right.
Fungi are amazing.
Fungi are amazing, and I think that's an end point to our discussion. And thank you very much to everybody who joined us today. Thank you guys for being my guests today. And for all of you out there, please keep an eye, and we will be back at some point over the summer. Thank you very much. Bye-bye, guys.
Thank you.
Thank you very much.
Bye.